Hi friends, welcome to my YouTube channel. Today I'm going to give you some idea about Raman spectroscopy. Before going into the details, let us try to understand what do you mean by spectroscopy. It helps to study the interaction between matter and the electromagnetic radiation like light as a function of wavelength or frequency. It provides information about the composition, structure, and properties of a particular substance. It can apply in chemistry, physics, biology, astronomy, as well as in environmental sciences. There are different types of spectroscopy, like infrared, followed by nuclear magnetic resonance, mass spectroscopy, X-ray spectroscopy, fluorescence, electron spin resonance spectroscopy, Moss-Bohr spectroscopy, absorbance spectroscopy, CD or circular dichroism, UV-Vis, Raman, followed by photoelectron spectroscopy, commonly known as XPS or UPS. Let us try to understand where this type of spectroscopy is generally applied. In the first case, that is IR, it is generally applied in identifying functional group in organic compound and to understand the structural analysis of many substances. And NMR, it helped to study the molecular structure, studying the protein conformation and metabolic analysis. On the other hand, mass spectroscopies are generally used in identifying unknown compound, studying biomolecules, analyzing isotopic composition, followed by X-ray, help to study the crystallography, elemental analysis, and material sciences. Fluorescence spectroscopy is generally used in studying biomolecules, fluorescence labeling, as well as drug discovery. ESR is generally used in studying free radical, characterizing transition metal complexes, followed by Moss-Bohr spectroscopy, help in investigating the chemical and magnetic environment of a specific element in materials. Absorbance spectroscopy help in getting the general analysis of substances as well as absorption characteristics. CD is applied in protein structure analysis, studying biomolecular interaction, followed by Raman is for identifying and characterizing materials, studying crystalline structures, followed by XPS or the UPS. It is used to analyze the surface and determining chemical composition and electronic states, followed by UV-Vis spectroscopy is generally help in detection of the impurities in a particular substance to determine the structure, elucidation of organic compound, and qualitative analysis. Out of the different spectroscopies, Raman spectroscopy is very much popular. It helped in measuring the inelastic scattering of the photon, providing information about the molecular vibration of a particular substance. It can be applied in identifying and characterizing the material as well as studying crystalline structures. The history of Raman spectroscopy started in 1923 when Austrian physicist Ismail predicted the inelastic light scattering. It was C.V. Raman and his colleague K.S. Krishnan who experimentally proved the inelastic nature of the light in 1928, and they received Nobel Prize in 1930. There was a huge breakthrough for the experiment of C.V. Raman with the invention of laser during 1960. Raman spectroscopy is a spectroscopic technique that uses the inelastic scattering of photon to study the vibrational, rotational, and other low frequencies mode of matter. It provides insight into the molecular composition and vibrational properties of a wide range of substances. In case of Raman spectroscopy, a beam of monochromatic light, usually from a laser beam, is shown onto a particular sample. The light interacts with the molecules in a sample and some of the light is scattered. The scattered light has the same wavelength as those of incident light. But a small fraction of scattered light has a different wavelength. This is called Raman scattering. When a monochromatic laser light interacts with a particular sample, the laser light can either be absorbed or transmitted or scattered. The scattering may be of two types: elastic scattering or the Rayleigh scattering, which is marked with blue arrow mark, and inelastic scattering or the Raman scattering, which is marked with red and green arrow mark. As we know that when a light is scattered off a sample, there are two possible outcomes. One is elastic scattering, which is also known as Rayleigh scattering, which is occur when the scattered light has the same energy as the light that initially struck the sample. This means that the elastically scattered light will be the same frequency, wavelength and color as that of the original beam of light. On the other hand, in case of inelastic scattering or the Raman scattering, it occurs when the scattered light has a different energy than the light that initially struck the sample. This means the inelastically scattered light will have a different frequency wavelength and the color than that of the original beam of light. Raman spectroscopy relies on the inelastic scattering of monochromatic light, typically from a laser source, as it interacts with a particular sample. When photons from the incident laser light interact with the molecule in the sample, some of them undergo energy changes. These changes manifest as shift in the wavelength or the frequency of the scattered light, resulting in what is known as Raman effect. The difference in the wavelength between the incident and the scattered light is called Raman shift. The shift is characteristic of the vibrational mode of the molecules in the sample. By measuring the Raman shift, it is possible to determine the frequency of the vibrational mode of the molecules. Raman spectroscopy is a non-destructive technique that can be used to study a wide variety of materials, including solid, liquid, and gases. As I mentioned, it is used in a variety of fields, including chemistry, physics, biology, and material science. So Raman spectroscopy is the analysis of the molecular vibration. As we know, molecules are not static entities but are constantly in motion. When a molecule vibrates, their chemical bond is stretched as well as bent, as I have shown in these pictures. And these vibrational modes are associated with the specific energy level. When a photon interacts with a molecule, it gains or loses energy, causing a Raman shift in its frequency. This shift corresponds to the energy associated with the molecular vibration, providing detailed information about the chemical composition and structure of the sample. So we have to remember that molecules have different vibrational level, and these are defined by the specific energy differences. When a particular molecule interacts with a particular photon, that molecule from ground state they may reach to virtual state. But the virtual state is a very short-lived unabsorbed state. Again, it goes down to the ground state. Here, frequency of the laser light is equal to frequency of scattered light. This is called your Rayleigh scattering. But in some cases, when a particular molecule interacts with a particular photon, it reaches to the virtual state, but they cannot go to the ground state, they remain up to the excited state. Here, frequency of laser light is greater than frequency of the scattered light, and it gives us the stock scattering. And this happen only in say 1 of the millionth photon, and this is considered as fingerprint of the molecule. In the third case is here, when a molecule which is still in the excited state come in contact with the photon and interact with the photon, they reach to the virtual state, then again it comes down to the ground state. Here frequency of the laser light is less than the frequency of the scattered light, and this is called anti-stock scattering light. Here scattered light has more energy than the incident light, and it creating anti-stock Raman. And here the main focus of the Raman spectroscopy is typically on the anti-stock scattering light, because at a room temperature these anti-stocks may happen as compared to the other two type of scattering light, that is Rayleigh scattering as well as stock scattering light. A typical Raman spectroscope has different components. Number one is light source, followed by sample chamber or probe, optical components, a spectrometer, followed by detector. Let us see a typical setup of Raman spectrometer prepared by Bruker. When a laser beam created by the laser reached to a sample through beam splitter and the focusing optics, it redirected to a particular filter, and through another optics it reached to the spectrophotometer system. That means initially the entrance slit, then the lenses will reflect it to the grating, and the grating splits the light into different wavelengths, and which are detected by CCD detector, and it transforms it to the Raman lines, which is commonly known as Raman spectrum. And here the intensity of the band corresponds to how many Raman scattered photon is detected by the detector at the specific positions. Regarding the light source that is used for Raman spectroscopy, I want to say that a monochromatic light source is the crucial component of Raman spectroscopy. Common choices include laser operating in the visible or the near infrared regions. Common laser sources include diode lasers, solid state lasers, and more recently fiber coupled diode lasers for portable Raman system. And the sample chamber or the probe is where the sample is to be analyzed, is placed or exposed to the laser light. For solid and the liquid, samples are often placed in the quartz or the glass cavities or the petri plates. In some cases, fiber optic probes can be used to collect Raman spectra directly from samples without removing them from their containers. The optical component, which is an important part of Raman spectrometer, are responsible for directing and filtering the scattered light from the sample. A beam splitter is generally used to direct a portion of the scattered light to the spectrometer while allowing the remaining light to pass through. Lenses of the mirror focus and collimate the scattered light, ensuring that it reaches the spectrometer entrance slit. A spectrometer is the crucial component for dispersing the scattered light into the various wavelengths of the spectrum and measuring its intensity at the different wavelengths. It is typically included at a diffraction grating or the dispersive element for wavelength selections. Modern spectrometer often used CCD or charge coupled device or complementary metal oxide semiconductor or CMOS detector for high sensitivity and rapid data acquisition. Next important component is detector. Detectors is responsible to capture the dispersed light and convert it into the electrical signals. CCD or CMOS detectors are commonly used for their ability to detect a wide range of wavelength simultaneously. Photomultipliers tube or PMTs are also employed for high sensitivity in certain Raman spectroscopy. Now regarding the data analysis software, so we have to remember that these are specialized software which is used to control the instrument, process the raw spectral data and generate Raman spectra. The software often includes features for baseline correction, spectral calibration and spectral interpretation. Beside that, some optional accessories are there. So depending on the application, additional component may be included such as polarization, optics to study anisotropic samples, temperature control devices or Raman microscope attachment for microscale analysis. Then for reference material, a reference material with well-known Raman spectra is often used to calibrate and validate the spectrophotometer's performance. Safety measures including laser safety goggles and interlocks are essential when working with the lasers to protect operators and ensure safety use. Depending on the sample type and analysis requirement, various sampling accessories may be used such as sample holders, cuvettes or probes. If you could able to handle the Raman spectrometer very accurately and you handle the software very perfectly, then you will get such type of clear picture as shown here and you will get good Raman shift with the intensity of your material. Such technique have number of advantages. So number one point I have mentioned, chemical specificity. Raman spectroscopy is highly chemically specific. It provides detailed information about the molecular composition of a sample based on vibration and rotation of the chemical bond. Each molecule has a unique Raman spectrum, enabling identification of substances even in complex mixtures. Followed by Raman spectroscopy is a non-destructive technique. It does not require extensive sample preparation and the analysis typically does not damage the sample. This is advantageous for analyzing delicate material and biological specimen. Raman spectroscopy often requires very small sample volume, making it suitable for precious or limited quantity sample. It can be applied to a wide range of sample type including solid, liquid, gas and biological material. It is also applicable to solutions, powder and surface analysis. Raman spectroscopy can be used to analyze aqueous solution as water does not interfere significantly with Raman signal. This is advantageous in biological as well as environmental research. Techniques such as confocal Raman spectroscopy and tip enhanced Raman spectroscopy offer high spatial resolution, allowing for the analysis of small regions or even single molecules. Raman spectroscopy can provide real-time information, making it suitable for monitoring chemical reactions, processes, and dynamic systems. In some cases, such type of spectroscopy can be used without direct contact with the sample, such as with fiber optic probes, reducing the risks of contamination. Lastly, Raman spectroscopy can be used in combination with other analytical techniques such as FTIR spectroscopy to provide complementary information and enhance the overall analysis. Besides that, it has number of limitations as Raman signals are relatively weak compared to fluorescence, which can make it challenging to detect trace level component or weakly scattering material. This is especially true for highly fluorescent samples. And to improve signal to noise ratio, Raman spectroscopy often requires longer acquisition time compared to other techniques, limiting its suitability for rapid analysis. The use of laser in the Raman spectroscopy necessitates laser safety precautions, including the use of laser safety goggles and adherence to the safety guidelines. Followed by fluorescence from the sample can interact with the Raman signal, particularly in organic material. Any strategies like resonance Raman can be mitigate these issues. From for some materials such as powder, it may be necessary to prepare a uniform sample deposition, which can introduce potential sample preparation errors. Then in the sixth point I have mentioned that Raman spectroscopy can be sensitive to the orientation of the sample, which may require multiple measurements to obtain a representative spectrum. Regarding instrumental cost, high quality Raman spectrometer can be expensive, limiting accessibility for some researchers. In complex mixture, spectral overlap can make it challenging to identify individual component of a particular substance. And lastly, Raman spectroscopy typically provide surface or near surface information, which may limit its applicability for analyzing substance features. For more information on Raman spectroscopy, I would like to request all of you to go through more YouTube video published by Brooker, Santera, Alpha, Invenio, and many more. And lastly, I would like to thank all of you for watching this video.